Good evening, ma'am.
Good evening, sir. You can start now, sir.
Good evening, ma'am.
Good evening, sir. You can start now, sir.
Yes, ma'am. Thank you. Good evening, my dear students. You are all welcome to the another questionnaire session 2023. This is Dr. Meenakshi Sundaram taking the biology classes. So the first question is there now. Already we saw it. How many questions is now? 80 question is there. Okay. We started remaining part of the question. The intestinal perforation is the characteristic symptom of, already you know, but already we saw it in the last class also, my dear, this part. Okay. The intestinal, what is that? Perforation. Okay. The intestinal perforation mostly occurs in which part, which cosmetic organism, like that they ask the questions. Pneumonia, plague, typhoid, and pertussis. You must know the exact cosmetic organism. These are the components, these components. First of all, you should know pneumonia. What is that? Pneumonia. Mycoplasma pneumonia. Okay, pneumonia. Okay, so it will not the intestinal perforation. Pneumonia is also one of the lungs disease. Just imagine lungs related. Here they ask the intestinal. Okay, this is also very, very important. Plague. Yersinia pestis. YER. Yersinia pestis. Okay, maximum it is disposed. It is also one of the bacterial disease caused by the which one, that rat. Okay, rat excreta, simply called as excreta. What is the meaning of excreta? Feces and urine. Okay, so this is also a very complicated. Plague is also one of the most dangerous diseases. It also occurs in the 1984, I think. And typhoid. Typhoid is already in our book. Salmonella typhic. Okay, 4 types of typhoid is there. This is the intestinal. This is called as intestinal perforations. Pertussis. Pertussis already in our book. Okay, triple C vaccine also. Triple C BCG. The vaccine also gives. Okay. Triple vaccine. Okay, vaccine. So that things also you must note it. And here the correct answer, the intestinal perforation mostly in the characteristic symptom of which one, typhoid only. Typhoid is one of the intestinal perforation only, intestine. Here pneumonia and plague, and also this one is called as dysentery. Plague is also dysentery oriented, and also fever and so on. So pertussis is nothing but the, what is that? Cough. Okay, that things also it comes. So exact answer is which one, see that one? The intestinal perforation is the characteristic symptom of the typhoid is the correct options. Typhoid is the correct option. Okay, media. So go to the next one. The incorrect statement, otherwise called as wrong statement. Okay. Here the incorrect statement, that is also called as wrong statement. Okay. The infection of Entamoeba histolytica, already known by Entamoeba histolytica, is also caused by which one, dysentery. Dysentery. Stomach, okay, related. Entamoeba, amoeba. Okay. So it is a protozoan. It is a protozoan. Parasite of human. Okay. It is a protozoan parasite of the human. Is it correct or wrong? Yes, it is one of the protozoa parasite mostly found in the intestine. Entamoeba histolytica, intestine. Okay, protozoa. It's a correct one. So it will not come to the wrong answer. Okay, second one, it is a characteristic by constipations and abdominal pain. Yes, already told you, intestine means automatically the Entamoeba histolytica mostly occurs in the child. Okay, childs or infants. So that's why they may give, which one, no. Sometimes they may give the other alternative choice. See here, it is a parasite in small intestine of man. No, it is not only the small intestine. So this is the wrong answer. Okay. Housefly acts as a mechanical carrier and serves as to transmit the parasite from the feces to infected person to the food and food product. Yes, it is the correct answer. So these are the 3 is very, very important. So it is also the Entamoeba histolytica is also a protozoan, and also it is a parasite of the human being. Constipation abdominal, as a result, a symptom of this one, when the Entamoeba histolytica transport through the food and water, what will happen, it will lead to the constipation and abdominal pain. Okay. And it is a parasite in small intestine. No, it is not a small intestine. Just to imagine, this is the wrong answer. So here the question is incorrect statement, the wrong statement. Housefly, already know housefly. Okay, act as a mechanical carrier. What is the meaning of mechanical? It carries the, which one, the protozoa in his leg. When it transmitted through the parasite from the fecal matter, affected person, the fecal matter, when it sit on it, what will happen, adhere on the leg. Okay, in his leg. So as a result, what it will be transferred through the food. When it touches the food, what happens automatically? The parasites or protozoan parasites also adhere on the food also. When you are consuming, what happens automatically? It will enter in your body. As a result, lead to what happened now. These are the problems. Okay. So here the wrong statement only they asked this. So this is the correct answer, my dear students. See the option. The endoamoeba histotic is the protozoan parasite of the human. Yes. So the first point is correct. Okay. And what about the second one? What about the second one? Just imagine. So it is characterized by constipation, abdominal pain. Okay. This is also already known. Housefly act as a mechanical carrier and thus transmitted the parasite from the feces of the infected person. Those who are already affected by the endoamoeba histolytica, that person excrete the excreta in the form of feces. It has lot of what is a parasite protozoan. So the housefly sit on it. What happened? Already I told you, and also transfer to the food. Okay, this is very, very important, my dear students. Go to the next one. Mark the add one pathogens. See here, mark add one pathogen. Don't forget, my dear students. Diphtheria, common cold, plague. Diphtheria pertussis. Common cold is also viral disease. Plague is also a Yersinia pestis. Typhoid is a bacterial disease. Okay. Okay. So which one is the correct option is also? Sorry. Here. This is also bacterial disease. Diphtheria also bacterial disease. This one is also called bacterial disease. So which one is the common cold is the correct option? Sorry, this one is not a correct one, because common cold is also cosmetic organism, which one? A rhinovirus. Rhinovirus. Common cold caused by the one of the cosmetic organism is viral. Other three are bacterial diseases. Already I told you, diphtheria pertussis, typhoid, salmonella typhic, plague, Yersinia pestis. These are all bacterial diseases. Common cold is a final. So as a result, what happened? Common cold already in our league. It will not cure. Once the foreign particle entered in the body, what happened? No, it will stay as it is. Suppose if you are having the immunity powers or not, whatever it may be, but they will not cure immediately when you are taking the tablets or whatever. 48 hours to what is that? 60 hours, maximum 3 days. When you are taking or not taking the tablets or any other antibiotics, when you are consuming, what happens? They will not support for the curable. Virus disease is not curable, only preventable only. That is very, very important, my dear students. Okay, see that option. The common cold is a viral disease. Others are, what is that? Bacterial disease. Okay, go to the next question, people. The typhoid fever could be confirmed by typhoid fever. Heavily identified which test is responsible. Those who are having typhoid, heavily confirm. See, the first test is called as what is that? Vidal test. Yes, the Vidal test is the confirmatory test for typhoid. Okay, don't forget. This is also so many times they ask the question. NEET 2023. Montague's test, Sick test, Dick's test. These are all not come for the typhoid. So this one is the correct answer. Remaining three or what is that? Wrong answer only. See the option here. The typhoid fever could be confirmed by the Vidal test. Don't forget, my dear student, this is very, very important. The Montague's test, what is that? You have to identify that things also. Sick test, Dick's test. So these are the tests. Okay, so you have to find out these are the tests where it will comes. That's just to go through the book. You will get the ideas. Otherwise, I will explain it the next day. Okay, Vidal test. Already we discussed the Montague's test also already we discussed. Otherwise, it will come back, I think. Okay, see the answer. The which of the following is an autoimmune disease? Listen here, autoimmune disease. Okay. Just to see the question paper, immediately you will attain which one in AIDS is the autoimmune disease. Okay. Is it correct? No. Asthma? No. Cirrhosis? It is not like that. But rheumatoid arthritis is the autoimmune disease. This is the keyword. Okay, this is the keyword. So answer is, listen here, rheumatoid arthritis. It is also joint pain. Joint pain. Okay, maximum it occurs in old people because the age is what happened now. Increase automatically the immunity powers also decreases. That's why it is related to autoimmune. Okay, old people mostly affected this type of things. Okay, media. So this is also a key point. Go to the next one. Rheumatoid arthritis in autoimmune disease. Okay, and go to the next one. See, this is very, very important, my dear students. So many times they were asked the question in this type. See, allergy, Montague's test. Already I told you, Montague's test. ELISA and hallucinations. Already we know about it. In this part, already we discussed so many times. Allergy is also caused by the one of the dust particle, isn't it? Dust particle. Those who are working in the where? The cement industries. Cement. Okay, industries. Cotton industries. Okay. So what happened? Dust also enter. As a result, what happened? Already you know about immunoglobulins, particularly allergy. It is related to which one? Immunoglobulin E. So the allergy is here. Immunoglobulin, where is the? Here is the fourth one. So see the option. See the option already you know about. See the option. Which one is the? The first one. The answer is 4. So the second or third one is the option. Correct answer. Okay. Then next one is Montague's test. Okay. Listen here, Montague's test. Montague's test means mostly they were used for the, suppose if it is, you do not know this answer, isn't it? Just lift it. What about the ELISA? ELISA already know about AIDS. So C is, the answer is second one. See, where is the C is second in this part? So this is the second answer, isn't it? C is second. So automatically you know about the third one. Second option is third one. So second option is automatically, what is that? Tuberculosis. Don't forget, my dear, TP. TP identification or the confirmatory test is called as Montague's test. Before slide also we saw it, the Montague's test is also, which one is that? So tuberculosis is the Montague's. Already you know, hallucination. Hallucination is nothing but what is the, you know, it is also related to the atropa pallidum. So the answer is which one? You know, easily you can identify. This is easy question, but it is not exactly. Okay. The easy question is that. Once again, I repeat for you this point. Allergy. Allergy is related to the immunoglobulin. Just imagine immunoglobulin E. Okay. This was secreted by the mast cell. Don't forget, my dear student, mast cell. Where is the mast cell is there? Mast cell already you know, but it produces the histamine protein. Okay, the histamine protein. It is an anti-allergy substances. So as a result, what happened? Your skin is also irritated, and also there are small patches like your skin. Automatically, what happened? You will go to the doctors for the remedy. That time, what happened? They will provide antihistamine drug. Antihistamine drug. Don't forget, my dear student. This is very, very important. So allergy is related to immunoglobulin E. Montague's test, tuberculosis. ELISA, enzyme-linked immunosorbent assay is the, what is that? Not a confirmatory test of AIDS. Let's listen here. Just it is the identification, whether the person suffering from AIDS or not, like that, they can easily identify. Okay, AIDS. Okay, but Western blot is the confirmatory test of AIDS. That is very, very important. Just listen here, my dear student. This is very, very important. So many times they were asked the questions, this part. AIDS normally caused by the organism. What are they? AIDS is also one of the important question is that. AIDS caused by HIV, human immunovirus. Okay. The test is called as ELISA, whether the person suffering from AIDS or not, just for identification. Identification. Suffer or not. Okay. Or not. Like that. But at the same time, Western blot method, just to note it down, Western blot method, just to listen here. This is the confirmatory test. confirmatory. Okay, confirmatory test for AIDS. Don't forget, my dear student, this is very, very, very important. Western blot method, so many times they asked. Enzyme linked immunosorbent assay, it is also AIDS, HIV. What is that HIV structure is there? Single standard RNA copy with single standard, what is that? RNA copy, I already told you. At the same time, 2 copies also, 2 copies. And also that a reverse transcriptase enzyme, reverse transcriptase enzyme. Don't forget, my dear, cryptase, 2 copies. Okay. So these things is also very, very important, my dear student. In this part, they may ask the question in this part also. Okay, next, ELISA. Hallucination already in our Atroba belladonna is one of the alkaloids isolated from the some plants, Atroba belladonna. Okay, see that. So the option is, which one is the correct answer? That. So this is also a NEET question, my dear student. In this year, we will ask. They may ask. We may expect in this part. See that allergy. Allergy is related to immunoglobulin E. Okay, immunoglobulin E. That is very, very important. Immunoglobulin, already I told you. Montague's test is a tuberculosis. The protein molecule, which protein molecule secreted at tuberculosis? The tuberculin is a toxic substance, otherwise called as toxin. ELISA is a test for the AIDS. Okay, it is not a confirmatory, not confirmatory. Just imagine, not confirmatory test. It is only the person suffer by the AIDS or HIV or not, like that. Confirmatory test is called as Western blot method. Western blot method. Don't forget, my dear student. Please take a notes, my dear. Please take notes. And hallucination is also related to Atroba belladonna. It's one of the alkaloids. One of the alkaloids isolated from them. Atropine. Is that atropine? Atropine is also isolated from the Atroba belladonna. Don't forget, my dear. It is used for the hallucination purpose. This part comes from drugs. Already we studied that. Drugs, isn't it? Tobacco, drugs, opium. Okay, papine, cannabis, sativa, all those things. That part, okay, don't forget. Go to the next one. Given below are 2 statements. Okay, and the first statement is, what is that, cancer-causing virus is called as oncogenic virus. Not automatically. Cancer-causing virus is called as oncogenic virus. Yes. Okay. The cancer-causing gene are called as viral oncogen. Is also a correct one. So the option is, which one is the correct answer? Both the statements are correct. Oncogenic virus. And cancer, the study of cancer is called as what? What is that? Oncology. Okay, media. Oncology. The science that deals with study of the cancer is called as oncology. The study of tissue is called as, what is that? Tissue. Histology. Okay. How will you identify the cancer cell? That is also one of the microtome is an instrument. Microtome. Don't forget, my dear student. This is very, very important. So many times they were asked the question. Tissue culture. How will you identify which one is the normal tissue? How will you identify the cancer tissues? How will you identify it? With the help of one of the technique is called as microtome. Okay. So the answer is correct. Both the answer is correct. On the oncogenic viruses, cancer-causing viruses is called as oncogenic viruses. Cancer-causing gene is called as viral oncogenes. So both are correct. The option is the third one is the correct one. Both the statement 1 and statement 2 are correct. Okay. This is the correct one. Remaining things are the wrong. Go to the next one. See this one. Cancer-causing viruses is called as oncogenic viruses, and the cancer-causing gene is called as viral oncogene. Reversibly, you have to put. Okay. Go to the next one. Cancer can be detected by. Cancer can be detected by. What are the methodology that were used for the identification of the cancer? Oncogenic things. Blood and bone marrow test. Yes. you know about the bone, okay, and also blood. Both are interrelated, isn't it? Okay, bone marrow produce blood component, so interrelated. Okay, this is the key word. MRI scan, magnetic resonance imaging. Sometimes they ask the questions expandable, expand. CT scan, you must note it down. This is also expandable. Expansion aspect, they ask the questions. All of this. Which one is the answer here? Yes, MRI can also is the sometimes. So those who are affected by brain tumors and some other cells, CT can also. This is also breast cancer, chest cancer, and so on. So you must note it down. What are the types of cancer is there? Cancer. There are 4 types is there. According to the position of the, okay, position and activity and physiological things. Okay. So what are they now? First one is called a sarcoma. The second one is lymphoma. Third one is carcinoma. The last one is called as multiple myeloma. Multiple means what is that? Blood cancer. Already know about blood cancer. Multiple alleles. Carcinoma, lymphoma, lymphocytes, bone marrow. Okay. Sarcoma and carcinoma. These are also. How it will be there? No, in the from the outside to inside. The outermost cell is the skin. Next tissues. Then bone. Then inside bone marrow. So likewise, the cancer is also classified. How will you identify? These are the methods. So the option is which one is the correct one? All of this is the correct one. Okay, go to the next one. Cancer can be detected by the blood and bone marrow test, MRI scan, and CT scan also. So all the 3 is correct. Go to the next question. The occurrence of the endemic species in South America and Australia is due to, what is that? Endemic species. Endemic species, South America and Australia. This is a keyword. Okay, media. The extension of these species from other regions. Extension of these species from other regions. It is not like that. Endemic means within the country only. The extension means outside, so it will not come. Continental separations. Continental separations. Okay. What is an, suppose take an example. This is the a place. Okay. So what happened, continental separation means it may be divided like this and also separated like this. What happened? The endemic species as it is present inside only. So the option is the C, B is the correct one. Likewise, America and Australia also separated by which one? Continental separation only. How many continents is there? 7 continents is there, isn't it? No terrestrial route to these places. No, it will not come. And retrogressive evolution. No. So which one is the correct option? The endemic species, endemic within the country only. Okay. The continental separation is the correct answer, my dear students. Go to the next one. The occurrence of the endemic species in South America and Australia is due to the continental separations. Okay, my dear. This is very, very important part. See, the next one is, what caused the onset of the puberty in male? Male, they told. Which one is the key point, key word here? Onset of the puberty in male. Okay. See, they increase the secretion of testosterone by testis. Puberty. No, not testis. Testosterone, it is one of the hormones secreted by, what is that? Testis. It is not a correct answer. Decrease the secretion of the testosterone by testis. Same is here the increasing word, here the decreasing word they have given. So both are is not possible. Listen here. The increase in the secretion of gonadotropic releasing hormone by hypothalamus is the correct one. Decreasing the secretion of the gonadotropic releasing hormone by hypothalamus is the wrong one. So what causes the onset of the puberty in male? it increases the secretion of the gonadotropic releasing hormone by hypothalamus. Where is the hypothalamus present in it? Hypothalamus, thalamus. Below the thalamus, hypothalamus is there. The hypothalamus secretes the hormone, gonadotropic releasing hormone. Okay, it is the essential for the onset of the puberty in male. Okay, so this is the concept, correct one. Others are wrong. So onset of the puberty in males is due to the increasing in the secretion of the gonadotropic releasing hormone by hypothalamus. Okay, my dear. So B, keep on practice. That is very, very important. Go to the next one. Pure line breed referred to already so many times we discussed this part. Pure line. If you want to get a pure line plantlets or some other things, you know, which one is the highest here? How will you get pure line means what is that? Heterozygosity, it will not come. Hetero. Pure means you need homo. So homozygosity only. So often it's already you know about heterozygosity, it will not come. What is my homozygosity? I already told you during the time of tissue culture. Okay, just imagine tissue culture. What happened? No, any one of the plantlets you have to you place in the agar medium, isn't it? For their growth, agar medium. Agar medium it contain nutrients, all those things, phytohormones, all those things. There pH and also temperature also you have to maintain. So as a result, what happened? No, the plantlets. What are the pods? Whatever it may be, any part of the plant body. Okay, you have to place it like this. Okay, what happened next? This one. Okay, the callus formation takes place. Undergo the cell division process, mitotic cell division, somoclonal and somoclonal. Simple somatic hybridization. Somatic body cell divide means somatic hybridization. The tissue culture is also one of the somatic hybridizations. You will get the single cell, uniformly cells you will obtain there. That is called as mass of cell is called as callus. Isn't it? After that, what happened? After this, what happened? You know, again and again, subculture. Already we discussed subculture. Okay, you have to take this one for the first mass of cell, isn't it, media? Mass of cell is there. Again, it will undergo the further division, further second culture, third culture, fourth culture, fifth culture, sixth culture. What happened here? What happened? The last one, 10th culture, what happened? It will be pure in nature. And only the homozygosity. So homozygosity only present in it. It will not any cross hybridization takes place because it is one of the asexual reproduction, asexual method. Okay, only one plant cell is also undergoes the continuous cell division, meristematic cell division, as a result of formation of callus. Callus also undergo the further subculture. As a result, you will obtain the pure culture. Okay, subculture. Okay, so that is called as homozygosity. It's also very, 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 very important. Pure line is a correct. Breeds referred to as the homozygosity. Example, tissue culture. Not only the tissue culture. So many parts is that. See that. Pure line breed referred to the homozygosity only. Homozygosity only. Go to the next one. PCR technique is best for, listen here, my dear. PCR is the technique used for or best for, they told. PCR is nothing but the polymerase chain reaction. Okay, my dear. Polymerase chain reaction, simply called as PCR. Okay. What is the main best for this one? Best for this one. Listen here. DNA synthesis. DNA synthesis, what is that? Single strand as DNA also produce the two strand. It is DNA synthesis. This is also replication. Okay, replication. They will produce the exact copy of the another molecules. One copy here, two copy. It is not a PCR technique, so it will not come. Protein amplification. Protein amplification, it will not come. DNA amplification. You know, but there is a DNA primer. There are so many markers also used there. If you want to isolate it, if you want to, what is that? Want to numerous quantity of the DNA molecule. Okay. Only through the way of DNA amplification only by the process called as PCR. For example, suppose take an example. This is the molecules 1 to 100. Here the molecules AUG, 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 like that it comes. AUG is denoted as methionine. Okay, starting codon. Okay, media. These are the important point. Suppose I need these DNA molecules only millions of copy, 1 lakh copy I need, they will refer which one, Okay, PCR technique. They need PCR technique. So this is the normal structures. Okay, here the primer is also ordered, DNA primer. Okay, and it will be produced number of copies, same component, same DNA molecule. How much is that? We cannot be able to calculate. Thousands and millions of copy you can put only through the PCR, polymerase chain reaction. That's why it is called as chain reaction. What will happen finally, the end of the result, the single, what is that, copy, you will obtain, how much is that? This much, by the process called as PCR. Okay, so the answer is the which one? DNA amplification only done by the PCR technique. Others, it's not possible. Just to note it down, my dear students. Okay, go to the next one. The PCR technique is the best for the DNA amplification only. Okay, so go to next one. Which technique used to introduce gene into dicots? Already we studied so many techniques also. Electroporation, electrophoresis, it will not come. TIE plasmid infection, it will come. Microinjection is will come. The answer, it will be these three only. Electrophoresis used for the separation of molecule. According to their molecular weight. According to their molecular weight. Okay, media. So this is very, very important. The technique is also already in our, you have to load it in this point, in also plus side and minus signals also there, and also buffer is also present in it. It can migrate towards the pole, positive, negative. As a result, you will get the band-like structures. This is called as electrophoresis. Electric field, we are separating the molecule, is called as electrophoresis. Electroporation. Listen here, my dear student. Electroporation. Okay. Electroporation means with the help of the electron, we are introducing the gene into the target tissues. Electroporation. Just you are putting the holes. Just take an example. This is the bacteria. Just imagine. Okay. Here the target cell is there. You are loaded with the micropipette. Okay. Take an examples of micropipette. Micropipette is also there. Just you loaded the gene. What are the genes we are going to introduce? Okay, you have to put inside injection like that. Just introduce this one. This is not a dicot plant. This is a dicot plant. See the TI plasmid. The dicot plant is mostly used for the TI plasmids only. Micro injection. These are the technique they were used where, biotechnology, gene technology, genetic engineering, all those things. But exactly in the dicot, okay, introduce which technique they were used. TI plasmid is the correct one. TI plasmids, tumor inducing plasmids. This is considered a plasmid. Extra chromosomal body present in the bacteria is called as plasmid. Likewise, the plant also, it has an extra chromosomal body. That is also one of the cell is called as plasmids. This is called as plasmid. It has a specific characteristics, resistant, all those things is there. Okay. This can be identified and also isolated with the help of the molecular systems already know about. After that is also, what is that? Cutting and ligazing, linking process also done. After that, it will be entered into the E. coli. Then it will be passed on to the plant. So this is the correct option, mate. Don't forget. Okay. Go to the next one. See the option. TA plasmid infection is to introduce gene in the dicot plant. You can expect this question in this year, my dear. In this question is also very, very important. NEET 2023, maybe. Okay. So go to the next one. Addition of antibiotics. Keyword. Okay. Addition of antibiotics and chemical engineering process help in. Okay. Help in what is that? Mixing and aeration of media? No. Maintaining anti-foaming condition? No. Maintaining aseptic condition? Antibiotics use, you mean what is it? What is the meaning of aseptic? Complete removal of the microbes. Okay, removal. Complete removal, aseptic. Listen here, two things is very, very important. Aseptic means there is no any microorganism present in the particular area is called as aseptic. Okay, when you are using antibodies, what happens? That area particularly, suppose take an example. So this is considered your hand like then here there is a wound is there. Why you are using the chemical antibiotics? Why we are using in this area? For example, only I am telling that it is not a correct one. Antibiotics we are using, you know what happened? Completely the surrounding, there are so many microorganisms present in it, isn't it? It can be removed by the antibiotics introductions. So it is maintained the aseptic conditions only. So this is the correct answer, my dear. Maintain the constant temperature and pH. No, it will not like that. When you are using the antibiotics in a chemical engineering process, help in the, what is that? Maintenance of the aseptic condition only, not for the mixing and the aeration of the media, maintenance of the antifoamy conditions, and also maintenance of the constant temperature and pH. Okay, so these are all wrong answer. This one is the correct one. Go to the next one. Sorry, go to the next one. Addition of antibiotics in chemical engineered process help in the maintenance aseptic conditions only. Okay, go to the next one. Which of the following is not an applicable of PCR? So see there where it is PCR essential for the DNA amplification, isn't it? But here they asked about the PCR is not applicable. Up to the answer. You have to think like that. PCR is not applicable. Okay, ELISA. Okay, diagnostic of pathogen, DNA fingerprint in paleontology. PCR is very, very essential for these three things. Diagnosis of pathogen. If you want to identify the COVID virus gene, how will you identify it? You take a small portion of the DNA molecule from the COVID virus. It can be undergo the amplification process. Number of amplification. What are the sequences? Nitrogen bases present in it. All those things you have to analyze. We may come to the conclusion, and also it enter into the electrocardiogram. What are the sequences is there? Then we may come to the conclusion. So it is very, very important. DNA fingerprinting, for example, any one of the culprit, if we want to identify, means what happened, you know, you have to take a samples from both. Okay, and then undergo the PCR technique. So then only you will get the identification. And paleontology is nothing but the science that will study of the fossils. Note it down, my dear. Fossils. Paleontology, whether it may be a plant component, whatever it may be. Okay, it is also related to carbon dating. You must note it down. Off-life period of carbon. You must note it down. Off-life period. Okay, life period of carbon. You must note it down. So the ELISA is the correct option, my dear. ELISA is the correct option. ELISA is not applicable in PCR. Others are PCR is very, very essential. Otherwise called as polymerase chain reaction. Okay, my dear. ELISA is not applicable PCR. Go to the next one. This one is assertion and reasoning. Okay. See here, the Taq polymerase. What is my Taq polymerase or is involved in the PCR technique? Okay. Yes. Taq polymerase is involved in the PCR technique. Is the correct one. It is highly thermostable that remaining active during denaturation phases. Taq polymerase is a thermostable only, but it is not denatured. It's a during denaturation phases, not active. Denature. It will not undergo the denaturation process. So it is the wrong one. So the option is, which one is the correct one here? Both A and R are true, and R is not is the correct explanation of A. No. And both A and R are true. Here the answer is R are true means what is it? Here the wrong answer is there. So it will not come. A is true, but R is false. So it is the correct answer. So both A and R are false. It will not come. So the option is third one is the correct one. Okay, media. Go to the next one. Taq polymerase is involved in the PCR technique. Taq polymerase is highly thermostable that remaining active during denaturing phases is the wrong one. Okay, go to the next one. Method of producing microbes and pest-resistant plant. This is the key point. Okay, method. Which method we are using? What is that? Microbes and pest resistant plant included. Okay. See here, now only we saw it. RNAs. Okay, RNA means RNA interference. I is denoted as interference. Okay. Yes, it is a correct one. Use of PT cotton already in our PT cotton toxin. PT cotton toxin already in our Bacillus thuringiensis, that is denoted as PT. Which gene is present in it? Cry gene. Okay. So the option is, this is also a correct one. So gene therapy, it will not possible for the, what is that? Microbes and pest resistant. Gene therapy mostly used for the, which one you know? Genetic disorder in the embryo stage only, embryonic stage only. Okay. So this is the wrong answer. So the option is which one you know? Both 1 and 2 only. So the last one. This is the confusing options. Okay, my dear. This is very, very important. Okay. So go to the next one. See the option. Method of producing microbes and pest resistant plant. Resistant plant only the RNAi. Ribonucleic acid. What is that? Inference. And also use the BT cotton and Bacillus thuringiensis toxin. We will get the microbes and pest resistant plant. That is very, very important. This is also a very, very important NEET question in this year. NEET questions 2023, particularly BT cotton, Bacillus thuringiensis. Gene is very, very important. Cry gene. Don't forget, my dear. Cry gene, sometimes they may ask the question, cry gene is present in where, you know, Bacillus thuringiensis. Bacillus. Okay, like that they may ask the questions. BT cotton also you have to find. BT cotton. Don't forget, BT cotton, Bacillus thuringiensis, and cry genes, very, very important. Okay, go to the next one. The main challenges for producing the insulin using rDNA, recombinant DNA. Okay, technique was getting insulin into mature form using bond, they asked. Okay. Already we saw it, this part also. When the pro-insulin converted into mature insulin. Listen here, this is the concept. Okay, already you know, but Eli and Lilly is the one of the, Eli and Lilly is the, what is that? Company, American company. Okay, American company. What they did, already you know about. The insulin also, it has a two chain is there. Okay, one is A chain, another one is called as B chain. Both are present. Okay, both are separately prepared and also joined with the help of which bond they ask. Same things also here also. Yes. Hydrogen bond is the, okay, hydrogen bond is the no. For example, one what is that? Chain is that this one is A. Suppose another one is considered as a B. In between, what happened? No, hydrogen bond is the no. It is not a hydrogen bond. And peptide bond mostly present in the protein. Okay, one amino acid united with the another amino acids. This is the concept. Ionic bond, no. Disulfide bond is the correct one. So listen here carefully. Two, what is that? Chain also connected with the disulfide bond. Okay, this is very one SH bond, sulfitryl or sulfide. So disulfide bond is the correct option, my dear students. Which one in a mature insulin? Okay, don't forget this. This was first introduced by the name of the country is called Eli Lilly, American company. Okay, go to the next one. The main challenge for the production of insulin using rDNA technique was getting insulin into mature form using disulfide bond only. It is a challenging because why, you know, the A chain as well as the B chain also produced by the different industries. This one is also produced by the one company. And another one is also, that is B chain is also produced by the another company. So these two are united with the help of disulfide bond only, SH sulfide. Then only will get, what is that? Mature insulin, mature. Okay. So one more is that the insulin contain how many peptide bond is there? Peptide, polypeptide is there? How many amino acids is there? That is also very, very important, my dear. A chain, B chain. A chain how much? B chain how much? You must note it down. This is NEET question. The bond is disulfide bond. Okay. That is, and also it is also prepared in the commercial aspect in the form of humulin. Okay. Humulin, that is very good. Why it produces humulin? Because normally those who are affected by the diabetes patients, what they did, they took insulin from the slaughterhouse. Okay. Maximum cow or buffalo. But sometimes they may collect the insulin from the where, that pig. So as a result, what happened? All is not accepted this component. This big component is also big. Insulin is not. It's also similar to that of the human insulin. So it is not accepted by the some persons. It causes allergy also. So they come to the conclusion which one is acceptable by the human being. They prepare by which one? No, commercially in the laboratory with the help of the company is called as Eli Lilly. Don't forget, my dear sir. This also year also comes. Okay, go to the next one. The how many variety of rice have been estimated in India? Okay. See that this is the direct question, my dear student. You must note it down. Very, very important. This is also one of the direct questions. 2,000, 20,000, 2 lakhs, 27. This is direct question, my dear students. In the India, how many rice is there? No, 2 lakhs. This is also a NEET question. Okay, my dear. Go to the next one. 2 lakhs variety of rice have been reported in India. Okay. Go to the next one. The technique that serves the purpose of yearly diagnostic include. Yearly diagnosis include, they have given both 1 and 2 also. Okay. If you want to analyze any one of the diseases, Elisa already know about AIDS, COVID. Okay, any other molecules. If you want to analyze, means what happened. These two only analyze. Serum analysis. What they will understand, what is by serum already we discussed. Okay, what is the meaning of serum? After blood clotting, you will obtain the yellow color fluid after clotting process. Clotting. After completion of clotting, you can imagine completion. Okay, completion of the blood clotting mechanism, you will get the yellow color fluids. Okay, yellow color fluid. That is called a serum. The serum used for the analysis of what hormone, protein, immunoglobulin, okay, and minerals. Okay, that things we can easily identify. We cannot use for the diagnostic purpose. Sometimes they may use the diagnostic purpose also. Serum is also used for the ELISA test. Okay. Here, what happened now? PCR is a DNA related one. This one is called as what is that? Serum related, antigen antibody related one. So the option is the fourth one is the correct one. 1 and 2 is the correct answer. It will not come. What is my serum analysis? Serum analysis mostly used for which one? These components, hormone, protein, immunoglobulin, minerals. These are all are not diagnosed included. Okay, diagnostic means what is that? It is also one of the supporting only, supportive. Okay, supportive test only, serum analysis. But exactly what is that? Diagnostic included. Which one? Technique they asked. It is not a technique. Serum analysis is not a technique. Okay, PCR is the technique. ELISA is also one of the technique. Okay. This is the correct answer. So others are wrong. Go to the next one is the last one. A technique that solved the purpose of the yearly diagnosis included PCR polymerase chain reaction and also ELISA enzyme linked immunosorbent assay. Okay, go to the next one. About what percentage of all existing transgenic animals are mice? This is the last question. At the same time, it's a direct question also there. Just imagine, this is very, very, very important. This time they may ask the questions. This time, definitely 2023, they may ask, may expect. Okay, my dear. See, 75 is there. Mice. Listen here, mice. Okay, mice is there. 75? No. Direct question, I told you. Direct. You should know, already known answer. Then only. Percentage they ask. Percentage means just you will answer it this one. 100%. Yes, correct. Like that. It is not correct. 50%. It is not correct. Exactly 5% less from the 100. So the answer is which one is the 100 answer? 95. Where the mice? See the about what percentage of the all existing transgenic animals are mice? Transgenic. 95%. That is very, very important, my dear students. Okay. So see the 91% of all transgenic animals are mice. That is very, very important. Once again, I repeat for your concept. Now you are studying so many questions. This is not enough, my dear students. First of all, you should note it down. Per day, you must attend 2 questions, morning and evening. See, morning 7 o'clock. Just imagine 5:30 to 7 o'clock. Okay, one test. And then also study. Afternoon 2 to 3. Again study. Like that, you have to practice it. Per day, you must study at least the last 15 days only is there, my dear students. 15 days only left. Okay. 15 days only left. How many chapters is there? Already know, but 38 chapters is there. Now only we are going to finish the 4th questions. Okay. 4 questions means 4 into 100, 400 questions we will finish it. We finished 400 each, 4 questions. Each one is have 4 option is there. So 4 into 400, 1600 questions already we studied. Just imagine. Okay. So each one it has 4 options, isn't it? So each one it has 4 options means only one is the correct answer. Remaining 3 is also. So the remaining 3 also you have to prepare the question. It is not possible now. So that's why you have to identify which one is coming the correct answer. Others are why it will not coming. That is called as deletion method, my dear student. That is very, very important, my dear. So in this part, biology, as biology, when you are answering, you have to practice it. Deletion method. Okay, deletion method. Deletion method also. Deletion means it will come or not. It will come or not. It will come or not. It will not come. It will not come. It will not come. It will come. Why it will come? That is also very well. Why it is not coming? That things also you should note it down. That is very, very important. Per day, at least you have to practice 100 questions, my dear students. Unit test, or you have to attend daily 100 questions. Okay. NCERT book, you should follow it. NCERT, isn't it? NCERT book. If you have NCERT book in fingertips, you will get 360 out of 360. No doubt at all, my dear students. Okay. So already you know the concept, but you cannot be able to pick up the answer. That is the problem. So that's the reason. So the practice it, you will get the exact answer, my dear students. Keep on practice, you will get succeed, my dear students. Please don't lose your hopes. Okay, my dear. Good luck. Thank you so much for the participation, my dear students. Today we will finish up to this level only. We will see the next questionnaire session. Thank you so much. Bye. Take care. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.